So my name is Alexander Hurbic. I am an R&D architect, as Alexei said, and today I'm going to present you a very interesting topic. It's called pushing the boundaries of biosciences with AI. What does it mean? It means that we have all many sciences like chemistry, biology, and they've developed to some extent, but with emerging of AI and machine learning technologies, we've got new possibilities and to tackle old problems with new power. And today I'm going to talk about this. So I present the group within the SoftServe. The group is called Science RDN, and we understand ourselves as cross-disciplinary scientists on the intersection of biology, chemistry, and artificial intelligence technologies. We have a 3-year history, and today I'm going to talk about 4 topics. First of them is a molecular generator. Second is binding affinity prediction. Third one is chemical reaction yield prediction, and the fourth is multi-omics data analysis. I will give you a time after each subsection, so please don't hesitate and ask questions when I will set a time for that. Okay. First of all, what do? We mostly work on drug development process. And as you know, drug development process is a large history. It takes 10 to 15 years. It's very costly. It costs $2 billion or more dollars per one drug to market. And it consists of the following phases. First is drug discovery. It's when the scientist in a laboratory or other scientific institutions, they're trying to understand what targets correlates with the disease. Targets means some protein or other biomolecule within your body, which is responsible for some physiological effect, and lead is a compound which binds with a target and modify its function. It's a drug discovery. It takes many years, mostly done in academia. Second phase is preclinical studies. The drugs are mostly tested for safety and efficacy. Safety means are they toxic, harmful, mutagenic, do they cause cancer, etc., etc. Efficacy means do this drug work? Does it work? Does it do what it should do? And the third phase is clinical trials. Clinical trials testing on people, first healthy people, then sick people, and then we have post-marketing FDA approval studies. Today, and all the technologies I'm going to talk about today, they are all related to early stage drug discovery here. It's early stage drug discovery mostly. Why these technologies are important and relevant? We want to identify novel targets, means a headache, and we know a target for headache, but maybe there is another target that we don't know, and it is better, it is more specific, etc., etc. We want to find it and then find a way how to modify its function. Second, we want to generate lead compounds faster, test it faster, optimize their structure and properties behind the wet lab without the synthesis. It will save us time and costs, and it is done through artificial intelligence now. And also another huge task is data management. Okay, now I'm making a pause, and do you have a chance to ask questions related to the last 2 slides? Do you have any questions? Okay, so I'll continue. As I said, today I'm going to describe 4 topics. I'll start from molecular generator. The molecular generator is a neural network, as you see here, and it knows how to propose new drugs. Here is the idea. We have a data set. We sampled the desired data. Then we check properties. Then we decode the structure of known molecules, correct their structure, check drug likeness, and check proposed leads with molecular dynamics. Here is a principal scheme. It's an ensemble of models. SMILES is a way of encoding of chemical molecules. It's our data set. In terms of machine learning, we have a data set of biologically active molecules encoded as SMILES. We put them into encoder. Think about autoencoder structure. You know that autoencoder is made of encoder and decoder. Encoder compresses the representation of a data into some compressed, very small embedding vector, and then decoder decodes it into a large vector. So what we do is we encode known biological molecules into embedding vector. Then we have a kind of embedding space, a space of molecules where we encoded their structure, and then we sample new embeddings. In this way, we generate new biologically active molecules, which were not previously known, but similar to existing ones. And we have a chance to discover new drugs in this way. Is this idea clear? I hope. Except that after decoding the molecule, we're checking for the structure correctness of the SMILES. This is done via with seq-to-seq spell check models. I will describe it on the next slide. And besides that, we predict different chemical properties, including activity, toxicity, and other things which are important during drug discovery. So we already have biologically active molecule with predicted chemical properties like molecular mass, activity, toxicity, and other things. It is what drug discovery scientists want. And here, I would like to give you a more detail on this sequence-to-sequence spell check architecture. It's a sequence-to-sequence model. Honestly, it's a LSTM. And on this example, you see that this SMILE was incorrect, has incorrect structure. Instead of C of a carbon, we should have a bracket here, a round bracket. And this mistake was fixed by this sequence-to-sequence spell check model. This work was completed and published in one of the Wiley journals pretty recently, around a couple of months ago. The work is called Toward Efficient Generation, because we generate stuff, correction because we correct them with a sequence-to-sequence model, and properties control because we control properties here with this separate predictor of unique drug-like structures. Unique because they are new, previously unknown, and drug-like because we believe that they are drugs. You can find the link to the publication here in this publication. I believe the slides will be available to everyone. Again, now I'm making the pause and you have a chance to ask questions related to this section.
Could you tell more about sampling new embeddings? What does this mean?
Great question, thank you. Sampling new embeddings, we took a known algorithm of space sampling. Actually, it's a vector space. It's called SMOTE, maybe you know, SMOTE. Okay, just to sum up, not digging deep into the technical details, it's a separate algorithm which subsamples vectors from a vector space. We give them like a example of a vector and it samples around it from a vector space.
Okay.
Thank you for the question. Somebody else? Raise a hand if you're sleepy already. Okay, I'll continue. The second topic, as I said in this agenda, is binding affinity. Binding affinity is a similarity of a ligand, this green stuff, to a receptor, this gray stuff. And the idea of our second publication is described here. So we know a receptor. On this image, the thrombin is depicted, and then we are testing this receptor against different ligand structures, and we predict binding affinity in the form of a inhibition constant. Inhibition constant is a typical way of measuring the binding affinity in a world of biology. Then we put this and this structure into the neural network, and we find the most affine ligand to this receptor. We do not predict the structure. There is a bunch of other methods for that. The uniqueness of technology is that we are using graph neural networks for this task. And here is an idea of how the ligand, which is a small molecule, is encoded into the graph. So imagine you have a chemical molecule. Here is trichloromethane is depicted. It consists of 1 carbon atom and 3 chloric atom and 1 hydrogen atom. And we encode all these atoms as features and weights. We encode each connection between atoms, each chemical bond with a set of features, and each atom with a set of features. The features are uniform for all bonds and all atoms. And the idea is that the values of our features reflect the properties of atoms and bonds. And if we encode the atoms and bonds properly, then we can describe the chemical properties of a particular molecule. Besides that, there are such intra and extramolecular forces as van der Waals and electrostatic forces and others. There are more than 50 of different forces. So we also trying to model them with attention mechanism. I believe you know what attention is. The idea is that attention is a kind of a external force to a molecule, and we also can describe it. This is how we encode the ligand as a graph, and the second, our second actor is a receptor. We also encode receptor as a graph, but the way of encoding is a little bit different. As you see from this picture, we are not interested in the whole receptor as a molecule because it's too complicated. Thousands and thousands and maybe even millions of atoms take part into interaction of receptor, but we're not interested in all of them, but mostly in intramolecular bonds. Intramolecular means not the primary structure, but the bonds which make secondary, tertiary, and other structures of a protein. We encode them as a graph. You see here that these are interactions between the primary structure. So it means that this spiral, this helix, interacts with a neighboring helix, and the bond is formed between the helices. And we encode this as an edge and nodes. And the same thing for other such interactions. They form a graph of a receptor. Why we do this? Because these connections, they affect directly how the ligand will bind to a receptor. Okay, so imagine we have a ligand graph and a receptor graph, and then we put all these two things into graph neural networks. So we encoded ligand as a graph, then we encoded receptor as a graph, and then we also encode the complex of a receptor to ligand as a graph in a spatial coordinates, and we put that into the neural networks. and then we concatenate the output of these neural networks and feed it to some value. This value reflects the affinity of a ligand to a receptor. Then we feed all this together, and in this way, we can predict this value later. This work was demonstrated at last year Neuraleeps. You see a poster here, and the idea that the receptor is encoded as a FASTA, ligand is a SMILES. We represent FASTA and SMILES via descriptors and a fingerprints, and we combine that into multi-layer perceptron and then feed to some value, either energy of Gibbs value or inhibition constant. And here are the results. If you're interested, we also published these results on arXiv. You can follow this link and read more about this technology. Okay, I finished this part. Do you have questions related?
This method looks like a way when we can build model on huge system when we have a lot of relationships. For example, rules of 6 peoples, and you can connect to president, so we can take a huge graph of people who connect one with another, and from this amount of links we can say some property about a people. Looks like this way.
Yeah, if you change the domain from the atoms and molecules and their properties to social networks, then yes, it's true. Social networks consists of people as nodes, and then people are somehow connected, interconnected via edges. And if you spot a huge set of people connected in some way, then you can say all of them are connected somehow. Then you can the property of this set. For example, it's a family related, or it's a, I don't know, believers in God, or it's a, I don't know, software engineers, something like that. But here we're talking about atoms and molecules and their properties. But the mechanism under the hood is pretty the same.
And in this molecular strategy, as I understand, you got another problems which looks like impossible connections. So you say that you have some mistakes in formula, so you should build some mechanism which fix your formulas and other things.
Yes, but it is related to a previous topic, a molecular generator. The generator here, it works with errors. This decoder, it makes SMILES with errors often. And we had a choice, just leave them as it is and just skip. And the other choice was try to fix the structures, which we did with the sequence-to-sequence translation, effectively. But that's another topic. It is relates to molecular generation. Okay, any other questions?
Yes, please. Can you please clarify when you explain how you coded the structure of the molecule, you exemplified on the 3-chlorine methane, but can you just clarify which other molecules have you tested? I mean, this is a for bioscience, this is a very simple structure, and like which bio compounds have you encoded and tested for in this approach? Thank you.
Thank you for the question. So we encoded and tested thousands and millions of compounds. I just gave this simple example so the idea can be grasped easily. Okay. As ligands, we encoded like such complicated molecules as this one and this one and other things. Any of them can be encoded as graph because atoms are somehow interconnected with each other within a molecule, and it perfectly fits the graph structure. I just gave this example just for you to understand it, the concept like easily. It is what relates to ligands. As for receptors, again, it was around 1,000 of receptors, different. Oh, yeah, they also can be encoded in this way. But yeah, there are many questions to this. Okay, honestly, to the receptor encoding, for example, you know that AlphaFold 2 recently produced this automatic folding of a protein into tertiary structure. And they also, as one of the steps, they encode the fastest sequence as a adjacency matrix. And effectively, adjacency matrix is almost the same as a graph because it's a flat structure where the connection between edges are shown as numbers, and no connection is as depicted as 0.
Okay, thank you.
Thank you for question. Any other? Okay, so I described these two topics, molecular generation, binding affinity, and now I'll start talking about chemical reaction yield. Chemical reaction yield, as you can understand from the name and title, it's how many products of a chemical reaction are we expect to have from these reactants. It relates to organic synthesis. Here is the idea. So we have some molecule, and we have here depicted two reactions, like adding 1,3-ethyl or adding ODPM rearrangement, and it produces two other different molecules. For one molecule, the yield is 50 to 60%. For other, the yield is 71 to 91%. It's experimental results, but what if we want to know these numbers prior to the synthesis, prior to deciding which reaction to choose? And this is the idea of chemical yield prediction. So using machine learning and AI and having a huge pile of data of performed reactions, their reactants and products and yields, we can predict yield then based only on the reaction conditions and the reactants. This was the idea of this work. And now I'm going to show you the model and the idea behind the concept. So we used a pretty complicated technique. Again, it was a message passing neural network, which is the graph neural network. It is capable of encoding of a difference between atom A and atom B in the form of a vector. So again, we encode all the atoms as all the molecules as set of atoms, and atom is a set of features. And then we compute this message, which one atom passes to another atom in the form of a feature vector. And we do this for reactants and products. Reactants is molecules which interact between them, and after the reaction, we receive products. So we encode reactants as this message passing feature vectors, and we encode products as these message passing feature vectors. Then we compute the difference between products and reactants. And the difference is what has changed in the structure of these atoms. Well, you all know that the chemical reaction is a simple rearrangement of atoms. According to the mass law, no new matter is created within a chemical reaction, and no matter is lost. The only thing is atom rearrangement. So we compute this rearrangement effectively. Then when they have to preserve the order of a chemical bonds, and in this way we are computing reaction encoding, and then we put it into another fully connected neural network, and then we feed it to the chemical yield. Chemical yield is this percent which you see under this arrow. It's the amount of a product which we expect to receive from this reaction. Yeah, that's it. This work is in progress. Like we are going to publish it in an upcoming month or two. Any questions related to this topic?
Do you consider the activation energy somehow?
Yeah, it's a known approach. I like during the literature review, I saw papers which they compute activation energies and then predict whether the reaction is possible at all. We just took another approach and another way. We omit this thing. We just predict yield. But yeah, the answer is no. In this work, we do not consider activation threshold. We just simplified the task to ourselves.
Okay, and the main idea beyond this study is just to sort of shorten this to save time of laboratory workers. For example, if you define some reaction to have yield only like 10% or 5%, then there is no reason of testing that reaction in real time in laboratory. Is
yeah,
that
of
right?
course. Yeah,
Okay.
you understood the idea.
Yeah,
Pretty good. I know many organic chemists in person, and they all complain that they have this retrosynthesis software, and it predicts the compound. What retrosynthesis does is, for example, the work of organic chemists is that they receive a desired compound, and their job is to synthesize it. And they build a synthetic path. It may consist of multiple stages, 5, 6, 10, 15, 3, something like that. And to build this synthetic path, they use the help of retrosynthesis tools. The retrosynthesis tools, you give the molecule to it, and then it proposes different ways of synthesizing this compound, but it doesn't say how reliable each path and how much yield can you expect from each stage. So this work, it's support work for this retrosynthesis task. It will design to give more information to chemists by simply telling you how much of a product is it expected to have at each stage. Then an organic chemist, by simple observation of yields on each path, it's much simpler to select the path. Okay, any other questions? Okay, thank you. So I described 3 topics, and now the last one is multi-omics. The fourth is multi-omics. So what multi-omics is? The idea of multi-omics, this is one of the samples of tasks. You know what the genomics is. Genomics is a set of nucleic acids of a person, maybe including mitochondrial genome, but yeah, for sure cellular. Then epigenome is a methylation profile of a DNA. Then transcriptome, it's a set of RNA transcripts from a genome, taking into account epigenetic modifications. It is received after RNA-seq, and then RNA, as you know, they encode proteins which are synthesized in ribosomes. So a set of proteins of a cell is called proteome, and then you have all the other metabolites within your body, which are, for example, adrenaline, testosterone, and other small molecules. They are called metabolome. So the idea of multi-omics is to combine all this information and predict some condition of an organism. In this case, this is one of the papers. They predict whether the person is healthy or sick, and it was for a cancer research. This is the overall idea. Here is an example of such a solution to such task. Right now, it's in the form of a Medium blog. You can follow the link and read more there. The idea is to combine the data I've just described. Here is data formats for this data and types of data, how they're collected. I'm not going deep into these details. Multi-omics is actually just a stacking of this single omics representation of a body, of an organism, and then applying machine learning, factorization methods, clustering, and other methods to this combined data to get some biological insights. For example, make personalized drug prescription, so we can predict whether this drug fits this individual or not. We can detect diseases. For example, it's early cancer detection and classify cancer type based on this information, which is very difficult having on some biomarkers in blood or urine. Then we can predict whether someone or something will survive after the disease and other things. Important application is industrial control of cell cultures. Imagine huge bioreactors where some culture of animal cells, it produces antibodies or something other, but very important biological molecule. Of course, this animal culture is genetically modified, but what we want to control the environment of this culture so that the outcome of this desired metabolite is maximized. This is our primary goal in this way, and we can compute such conditions by applying multi-omics data analysis approaches to this culture collected data. So we collect the genome, transcriptome, proteome, and other data about the culture, and then we can derive the best possible conditions. Here's the major result of this study. After multi-omics analysis, again, this is a distribution of a gene mutations of the data which participated in the research, and we see that one of the mutations clearly outstands a group of others and also have the largest possible weight, 1. These weights are normalized between -1 and 1. So the importance of this mutation is critical, very important, and it was like a very cool result of one study where they, by applying this multi-omics analysis, they discovered this immunoglobulin heavy chain variable region, IGHV, which is now the major clinical marker for the chronic lymphocytic leukemia. It's one of the types of cancer. So they simply, after performing this analysis, now they simply can spot this marker in the blood of a person, and then say with a 100 probability whether a person is sick with chronic leukemia or not. Well, it's a huge discovery, as you can understand, just from a data analysis. It affected lives of millions of people. And also there is a publication for that. you can click on this link and like check. It was like recent publication for discovery of this marker, and now it is applied in different studies and in clinics. So it is everything I want to tell you today. Please ask questions if you have any.
Someone asked you in chat.
Okay, I see. Okay, so do you there in software indeed have some deliveries related to what you show here? RBD to small molecule binding intimation, chemical reaction. What do you mean by saying deliveries? Did we deliver some projects?
Yeah, was like that. So is that something a person in Ukraine can work with, kind of?
Yeah, of course. Amount of
In
people,
software?
it's around 5 people in the group now. We delivered more than 4 projects to the time, and I can show you one of them. It's we've built a genetic data platform for COVID-19 patients based on multi-omics technologies. Well, it was a single omic, honestly, but it doesn't matter. It was a real-world project.
Yeah, interesting. So at least there are 5 people in Ukraine who can work in this area. That's nice to know. But you know, if 5 people for all Ukraine is just so little, so it's almost impossible for a new student, for a kind of a graduate student from some university to plan development of his career into that. So it seems to be so marginal.
The field is, of course, you understand, all of us understand that this field and this area is much more narrow than a big data, for example, or a computer vision, for example. And also it's a hard path because biological data is messy. It is often inappropriate, wrong, broken, outdated, and also it's almost impossible to take into account all factors which affect the organism, because even radiation and electromagnetic fields, they affect the body, and sometimes not all effects can be observed from the data. This is the difficulties. But the more difficult the path, the less people you will meet there.
Yeah, but also it's harder to earn money, actually.
Yes, of course, but we are not looking for easy ways.
Yeah. Also, another question. I've been watching YouTube for different machine learning stuff, and I found a person who has been actually given. It's a famous in Russia, some famous founder of some organization, and he was given lessons for institute, so-called Institute of Bioinformatics. Are you familiar with that?
Probably. Gelfand.
No, that was not Gelfand, that was a Russian one.
The Russian one, he is a Russian.
No, I can actually find his name, but I need to see my records.
Okay, doesn't matter. You found the person and what?
Yeah, it's just very close to your sim. So probably you should have been acquainted with that Institute of Bioinformatics. They just do, including what you actually depict here. And the name of the person is actually Grigory Sapunov.
Sapunov, okay.
So maybe if you are not familiar, maybe you can look. Okay, thanks. And one more question, actually, regarding multi-omics. The thing, even when we say about this Google framework, which shows now good results in folding proteins, it seems to be kind of on the very edge of existing possibilities for complexity. So probably multi-omics task as whole is probably beyond available now computing resources. So can you
comment on that?
Can you speculate on that?
So first of all, AlphaFold is OpenAI technology, as far as I know, not Google.
Oh, yeah, sorry.
Second, multi-omics, yes, it is huge. It is very hard to put it into any computational device, but current GPUs, they are thousands of gigabytes of volume, and everything is possible now. Also, we have quantum computers now. They are pretty weak, but anyway. So the task is doable. You can check the analysis performed in the article. It is actual analysis on actual data. The problem is amount of people in this data. So in this study, only 200 people participated. If we want some population, a large population, then of course, we will need much more resources to do that. But right now, we haven't, I think, like mankind haven't collected such large amounts of data. And I believe that with time, the technologies and the hardware will evolve, and we will be able to tackle these tasks. And also, AlphaFold is a protein folding thing. It predicts what tertiary structure a protein will have, and multi-omics is a little bit different. AlphaFold predicts the structure of a protein, coordinates. It's so you give FASTA. It's a primary structure of a protein, just amino acids, sequence of amino acids, and AlphaFold tells you the coordinates of a protein folded. Folding is it's a very difficult process which takes part in cells, but the outcome is statistically can be predicted, and this is what the AlphaFold does. Multi-omics is a little bit different. It just takes different aspects of an organism to derive some insight. For example, like in this case, it was a mutation which correlates with the presence of a disease. So the tasks and domains are a little bit different.
So we cut some edges actually here in this multi-omics task.
Not edges, we just don't pay attention that much to the structures. We do not pay attention to structures at all. Sufficient is only to have reads. It's a long topic. RNA-seq and it's a long topic, but we work on another level. We do not cut edges. It's just another perspective of data.
Sorry for interrupting, but due to the time, yeah, we need to continue our conference.
Yeah,
anyway, you can continue communicate with Alexander in social networks. I think that's not a problem. Thank you, Alexander, for bringing up such an exciting topic of AI and bioscience symbiosis.
Okay, thank you for questions and for interest. See ya, maybe.
Thank you.